Our today's topic is fossil form and function, and in which we will be discussing that what are the different ideas in the fossil form and function that we will be discussing in the coming lectures. Now, if we see the common species, the species are, as we have discussed in the earlier lectures, they themselves are very different from each other. The organism within a species, they are different. So when the same species organisms are preserved in the form of fossils, their fossils may be different as well. So fossil species are identified according to their external form. So if we have to see a particular species, we will see in a fossil form, we will see their external form, because that's the only thing we are able to see. And this is termed as the morphological species concept. So biological species concept, that was quite complex, right? So we will be able to see their DNA, RNA, organ structure, the bone structure, everything, because we have the organism. But in the case of fossils, we are only able to see their appearances. So that means this gives us a more wiggle room, right? So we only see the morphology. So the chances of error, they are much more. And the variation in the form include the normal level of individual variation. Of course, the same individual in the same species will have the variations, and we can find those in the fossil as well. Now, the fossil species might have other variations as well. The other type of variation in the form of fossils include geographical distribution. So if an organism is living on the equator as compared to the same species of organism living near to the poles, so there will be some changes in the size. The organism living in this, maybe an animal living on the equator will having the less size, right? But the organism which is living near to the near to the poles will have more size due to some thermoregulatory problem. And the sexual dimorphism, male or female, and male normally, for example, there are some males of species, of the same species, they have larger size than a female. And so is the case in case of peacock. Peacock has very beautiful plumage in case of males, but female is not as attractive. And there are different growth stages as well. And a growing organism might have different structure and might have different morphology as compared to the organism which is, having higher stages of growth, for example, an adult organism. And ecophenotypic variation, there are some changes which are given by the environment, right? So that is also, change the fossils as well. If organism is living in some place where there is some sort of, there are some changes in the environment, they can also change the structure of that organism or the fossil. Now, what is allometry? The fossil species may show allometry, or changes in relative proportion during growth. So that means there are some organs which are growing at one proportion, and the other organs are growing at other proportion, right? So that is called allometry. Allo means the different. Metry means that there is variation in their proportions. Their measurements are different. So now the development and phylogeny, these are also quite related to each other. The development of an organism may give some evidence about the phylogeny, right? So if you see the embryos of different organisms and how they, during development, they change, that also gives you a window of observation that how an organism have evolved throughout the history. And the changes in the developmental rate and timing, which is heterochrony, may affect evolution. Hetero means the different, chrony means time. So that means the changes in the timings also give some insight about the evolution. Now there is the new Evo-Devo perspective. Evo from the evolution, and Devo means the development. So evolutionary developmental biology perspective, right? So this is science of evolutionary development biology. It shows that how certain developmental gene control and fundamental aspects are formed. For example, the symmetry, the front-back orientation, segmentation, and limb form. All of these things which are characteristic of an embryo, these are, evolutionary developmental biology perspective. These tell us that how actually an organism have evolved in its history of evolution. So, inferring function from ancient organism is not a very easy task. It is difficult. And, when we do this, there are various methods. For example, comparison with the modern analog, or the biomechanical testing, or circumstantial evidence. So, for example, if we talk about the modern analog, for example, we do have the elephant in today's world. What are the ancient organisms that are related to the elephants, and we are, curious for them? We do see them fossilized, or we see them preserved in the earth layer, and we do know they existed. Though I am talking about the woolly mammoths, which are very much related to the elephants. So modern analogs provide exact parallel with some fossil organisms, and more often they provide only principles or rules, right? So if I see the locomotion of a common elephant, I might infer that how a woolly mammoth was walking and what was the gait of that woolly mammoth. And there are the biomechanical models. These can be used to assess how a design of an ancient organism matches the hypothetical forces against on it. For example, if I am talking about, again, the woolly mammoth, if he or she is walking, if it is walking, then the effect of gravity, what would be the effect of gravity on such a huge organism, or maybe the dinosaurs, which are very, very massive, right? So how did they cope with the gravity, and what was their rate of walking? Were they able to run, or what was the median speed with which they were walking? So this is, we can do this by the biomechanical models. And these can be used to assess how a design of ancient organism matches the hypothetical forces which are acting on these. And then what are the biomechanical models? The circumstantial evidence, such as enclosing rocks, right? So there are some... All right, so what is the circumstantial evidence first? The circumstantial evidence is the evidence that you find related to the fossil. It is not exactly pertaining to the fossil, but it is somehow along the fossil. So, for example, the analog enclosing rocks, the fossil is enclosed in the rocks. So, study the rocks, you will know a lot about the conditions in which the fossil organism was living. And the other fossils along with it, which are called associated fossils. Trace fossils, for example, some feces or other things of the organism whose fossils you have discovered. And you can also study the fossils closely to see that what are the forms or functions that particular organism may have, right? So all of these things can add considerable information on fossil function. So all these are the things, these are the topics that we will be covering in the next lectures. So these will be a basics of our coming lectures.